Hi everyone, welcome back to my A-level biology lecture series. In the previous lesson, which is meiosis part 2, where we explore meiosis 1, the reduction division. In this lesson, we will continue to explore meiosis, which is meiosis 2, the separation division. Let's get started. Meiosis 2 is the second division of meiosis following meiosis 1. It is important to know that there is no interface and also DNA replication occurs between meiosis 1 and meiosis 2. And the number of DNA remain unchanged, ensuring the chromosome numbers stay haploid throughout. And similar to mitosis as well as meiosis 1, it consists of 4 stages, and these 4 stages are prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis 2 play a very key important role to complete the meiotic process by separating sister chromatids at the centromere and to ensure that each resulting cells remain haploid and contributing to the productions of 4 genetically distinct haploid cells or spore in plant. Previously in meiosis 1, from 1 diploid cells, and after the end of meiosis 1, 1 diploid cell is divided into 2 haploid cells. But these haploid cells, each chromosome consists of 2 sister chromatids. So therefore, in meiosis 2, we want to separate sister chromatids and from 2 haploid cells into 4 genetically distinct haploid gametes. So let us start with prophase 2. So in the cells of prophase 2, we can see that, okay, the chromatid starts to condense to form chromosome, and also this pair of centrosome start to separate and move toward the opposite pole, and these nucleolus disappear, nuclear envelope disintegrated. So this is exactly or very similar to the prophase during mitosis. So per cells, now the number of DNA is 46, and the number of chromosome per human cells, because this is haploid, so the answer is 23. So what are the behavior of chromosome? So we know that signature event of prophase. So during prophase 2, chromatin fiber condense to form shorter and thicker chromosome, observable, okay, under a light microscope. And each chromosome is composed of 2 sister chromatids attached at the centromere. A kinetochore, again, a special protein complex at the centromere formed at each chromatid so that spindle microtubule can attach to this kinetochore and push this chromosome to the metaphase plate in metaphase 2. Centrosome. So centrosome duplicates for meiosis 2 typically occur during interkinesis. So interkinesis is not in our syllabus. Interkinesis basically means between meiosis 1 and meiosis 2, a very short resting stage between meiosis 1 and meiosis 2. And during prophase 2, the pair of centrosomes separate and migrate to the opposite pole of the nucleus. So spindle microtubule begin to assemble from the centrosome to form spindle. At the onset of prophase 2, the nucleolus gradually disappear and nuclear envelope begin to disintegrate into small vesicle. No event associated with plasma membrane, and the plasma membrane remain intact during prophase 2. Next, let us look at the metaphase 2. So from the diagram, we can see that chromosome now align randomly at the metaphase plate. Okay, and each each centromere, okay, or kinetochore centromere is attached to the kinetochore microtubule, okay, from the centrosome at the opposite pole. And you won't be able to see a nucleolus because nucleolus is completely disappeared, and also nuclear envelope is completely disintegrated. So at this moment, how many DNA per human cells? Answer is still 46 DNA. And the number of chromosomes per cell, because this is meiosis 2, the cell is haploid. So the answer is 23 chromosome per human cells. So during metaphase 2, all chromosomes are aligned individually. This is chromosome, not homologous pair anymore, because we know that during metaphase 1, homologous pair, or we call them as a bivalence or tetrad, arranged at the metaphase plate. However, during metaphase 2, all chromosomes are aligned individually along the metaphase plate. And orientation of each chromosome is random, leading to the independent assortment of sister chromatids. This won't happen during mitosis because sister chromatids are identical to each other. However, due to the crossing over, the sister chromatids are no longer identical to each other. So how the arrangement, okay, or the alignment of these chromosome during or at the metaphase plate during metaphase 2 contribute to genetic variation. And for each chromosome, the kinetochore of the sister chromatid attached to spindle microtubule originating from opposite pole. So now for each chromosome, it has two kinetochores, and these kinetochores attached to two sister chromatids, both from the opposite centrosome. So how about centrosome? So the centrosome have arrived at the opposite pole of the spindle, and spindle microtubule are fully formed, and they attach to the kinetochore of each sister chromatid, and each chromosome receive two kinetochore spindle microtubule. Nucleolus has completely disappeared. A nuclear envelope is fully disintegrated. No event is associated to plasma membrane, and the plasma membrane remain intact during metaphase 2. How about anaphase 2? The signature events of the anaphase 2, we can see that sister chromatids move towards the centrosome away from the metaphase plate. And due to the attachment of the spindle microtubule to the centromere, so therefore centromere always move first, give us the signature characteristic of anaphase 2. So if you compare to anaphase during mitosis, there is the event is exactly the same. Okay, so how many DNA molecule per human cells? The answer is still 46 because there is no division of the cells yet. And now you can see that the number of chromosomes here, remember the chromosome now refer to the sister chromatid. And due to the separation, okay, the divisions of the centromere, so now sister chromatids are separated. So each sister chromatid is counted as one chromosome. So what are the behavior of the chromosome? So sister chromatids are separated. Why? Because of the centromere divide due to the shortening of the spindle microtubule. So the sister chromatids are pulled towards the opposite pole of the spindle by the shortening of spindle microtubule. And the centrosome move first because spindle microtubule are attached at the centromere region. How about centrosome? So the centrosome remain at the opposite pole of the spindle, and the spindle microtubule shorten, pulling the sister chromatid apart by separating them at the centromere. Why? Because the centromere is divided equally. And this to ensure that each chromatid move towards the opposite pole of the cell. The nucleolus remain absent during this stage, and nuclear envelope remain disintegrated throughout the entire anaphase 2. Again, there's no event is associated to plasma membrane, and the plasma membrane remain intact during anaphase 2. How about telophase 2? So during telophase 2, each sister chromatid reaches the opposite ends of the cells. Nuclear envelopes start to reform, and due to the sister chromatids uncoil to form chromatin, and nucleolus start to reform as well. And the spindle microtubule is degenerated, and cleavage furrow is formed, so that all two begin the cytokinesis. So at this moment, the cells is not divided yet. So the number of DNA per cells is 46, and the number of chromosome per cells is 46. And against the number of chromosome meant the number of sister chromatid here. So at this moment, per cells, we still have 46 sister chromatid. However, at the end of the cytokinesis, where the cells is divided, the completion of the cell division, then each cells contain 23 DNA and 23 chromosome. So behavior of the chromosome, sister chromatids now reach the opposite pole of the cells, forming the basis for two separate haploid nuclides again. And the sister chromatid begin to uncoil, become thinner, longer, and less visible as they revert back to the chromatin fiber. During telophase 2, centrosome remain at opposite pole of the cells. The spindle microtubule are disassembled or broken down. Nucleolides are reformed within each new haploid nucleus because of the sister chromatid starts to uncoil to form chromatin. Nuclear envelope is reassembled or reformed around each set of sister chromatid at two ends of the cells, and a cleavage furrow formed leading to the cytokinesis and the separation into two haploid daughter cells. Next, let us look at cytokinesis and chromosome status following meiosis 2. So after telophase 2 of meiosis, cytokinesis completes the formation of new daughter cells. So in animal cells, a cleavage furrow forms at the equator of the cells. So this furrow deepens as contractile protein constrict the cell's membrane, eventually splitting the cytoplasm into two separate daughter cells. However, in the plant cells, vesicle from the Golgi apparatus fuse at the center of the cells to form the cell plate, and each vesicle contains cellulose molecule, so that this cellulose molecule can form the cellulose cell wall. So this cell plate gradually develops into a new section of the cell wall, effectively separating the two daughter cells. Okay. And at the end of cytokinesis, follow meiosis 2, we were going to have 4 haploid cells. And each of the cells is genetically unique due to crossing over and independent assortments that occur during meiosis 1 and also meiosis 2. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comments section. See you in the next lesson and have a great day.